it's good to join again, both for the Vault and for the MVIF. And as the title says, I think we'll talk about something a little bit different during this talk that we've been thinking about a lot recently, which is viral interactions in microbial communities generally. And just as a brief reminder, it's easy for phage in particular to float around microbial communities either as free viral particles or as integrated phage that gradually degrade into prophage. And these change bacterial phenotypes in ways that we don't often think about. Of course, phage can kill bacteria when they become lytic, but there are subtler effects as well. You can have different lineages of closely related phage that compete with each other within the same, sometimes within the same underlying bacterial population. Integrated phage can change phenotypes of their host bacteria, whether that's differences in nutrient utilization, whether that's differences in interbacterial interactions, or both phage and eukaryotic viruses can change how bacterial and non-viral members of microbial communities interact with a host, either directly or in immune-mediated ways. So it's important in considering microbial diversity and microbial phenotypic diversity generally to be able to take advantage of, to consider phage and eukaryotic viruses, as well as being able to see them and characterize them in microbial communities in the first place. So like I mentioned, we've been thinking about this more recently as part of the Human Virome Program, or HVP, which was launched at the beginning of this year by the NIH. So it's still new-ish at this point, I'd say. I am delighted to run the coordinating center for the HVP along with Owen White and his team at the University of Maryland. So we're gearing up to make sure that data and resources and documentation and information from the project are appropriately shared. The HVP overall consists of 5 large virome characterization centers, or VCCs, that are tasked with generating body-wide human viral profiles, as well as 5 tools and protocols projects and 5 functional studies, the former of which are developing better experimental or computational protocols for studying viruses, and the latter of which are each doing different biological deep dives into specific virome biology. Something that we've worked on as a lab that does a lot of computational methods development is improving viral profiling from metagenomes and metatranscriptomes. Viral isolation and culture is even harder than what we've been hearing about for bacteria. And there are now a number of really good VLP, virus-like particle enrichment protocols, that can generate virally enriched metagenomes or metatranscriptomes for RNA viruses. They can be highly variable in effectiveness. They can enrich or deplete different viruses. But together with large-scale assembly programs, these have generated just recently really nice catalogs of viral metagenome assembled genomes, viral MAGs. We know that for any given sample, these will undersample the available diversity, viral and bacterial. So for any given metagenome, only a fraction of reads assemble, only a fraction of those are viral. We don't have good ways for checking viral bins that are nearly as good as those used by, say, CheckM for bacteria. So in order to drive programs like the HVP and to help preserve viral genomic and phenotypic diversity alongside other microbes, we wanted to better detect and quantify viruses in normal metagenomes and metatranscriptomes, since there are tens of thousands of these generated without the benefit of VLP enrichment. So we put together a package called Baklava. Like most of our tools, it has a complicated acronym, but the 3 main components of Baklava all focus on different ways to detect and quantify viruses in metagenomes and metatranscriptomes. The first focuses on nucleotide matches by building unique markers for viral nucleotides, building on technology from our Metaflan platform. The second component, because viruses evolve and diverge so quickly, focuses on more remote homology using amino acid protein matches, building on technology from our Human functional profiler. And the third component uses a neural net deep learner to classify contigs as viral or non-viral, very similar to what is done from traditional assemblies at this point. And then at the end, Baklava integrates these 3 types of information to get a viral community profile per metagenome or metatranscriptome. To talk just very briefly about some of the methods, we integrate as many viral MAG databases and isolate genome databases as we can get our hands on. We cluster these at approximately the level that the ICTV recommends for viral species, creating viral genome bins, which are analogous to species-level genome bins that we in Nicholas Agata's group use for a lot of our tools. Viruses, again, because they evolve and rearrange so quickly, require an extra step of superclustering to capture rearrangements, so cases in which the same viral species may not be whole genome homologous. And from here, we assign taxonomy and identify nucleotide and protein markers, where our nucleotide markers are a series of adjacent short k-mers, 100 base pair sequences, that are sufficiently unique to each VGB that when we see them, we can identify, or we can be confident that we've identified that VGB. So very similar to what Metaflan does using genes for bacteria, but now using arbitrary chunks of genome for viruses. And this allows us to achieve particularly high precision and recall for recognizable viruses, and then reasonable reasonable recall and precision for novel viruses. So if we look at the comparison between known, unknown, and non-viral sequences, we're doing great for known viruses in both precision and recall. We're pretty good at finding new viral sequences, and we're also pretty good at eliminating non-viral sequences in a variety of different synthetic mock communities. So to give you a sense of how this looks when used for one particular slice of human biology, we've profiled a set of several hundred metagenomes and metatranscriptomes from the second phase of the Human Microbiome Program, or HMP2. And very briefly, these follow 100 or so individuals longitudinally for 1 year each, collecting paired metagenomes and metatranscriptomes from stool about every 2 weeks. And we're contrasting ulcerative colitis and Crohn's disease phenotypes in IBD with non-IBD controls. IBD has been extremely well studied with respect to the rest of the microbiome. And we see things in the viral profiles like reduced diversity during disease activity that exactly parallel what's been seen for many, many years in bacteria and other microbes. Interestingly, in the viruses, this is true even in the absence of disease activity. So just in the contrast between IBD patients and control individuals, we see reduced viral diversity, which is not true for the bacteriome. Usually it takes a flare of inflammation to knock down the rest of microbial community diversity. Similar to the bacterium, though, IBD destabilizes the virome composition. So IBD patients tend to have more variable viral diversity, both between each other and within their own microbiomes over time. The way in which these viromes personalize over time is actually quite diverse, though, and this is less true in the bacterial community. Some individuals carry almost entirely unique viruses, so their viruses don't show up in anybody else. Other individuals carry almost only viruses that are highly prevalent in the rest of the population, and everything in between. So as has been seen in other contexts, we're seeing even more personalization in viral community carriage than in the rest of the microbiome. And then finally, we can use statistical methods to associate specific viral features with IBD diagnosis or activity. We and many others now have done this for bacterial and archaeal portions of the gut microbiome historically. We have a statistical package called Maslin that does this, that again, I won't talk about today, but we just released a new version with some updated statistical models. More importantly, for the viral communities, we're able to use exactly the same statistical model, incorporating longitudinal information, exposure information, biometric information, technical information, and identify specific viral features that are enriched or typically depleted or lost during ulcerative colitis or Crohn's disease. And just by glancing at the top slice here, many of these are crassphage, other phage. Crassphage are typically Bacteroidetes phage. Many of them are unclassified. And here we've actually showed fewer of the unclassified viruses. Typically, when we go looking at just about any phenotype and just about any sample set, most of the viruses that we're seeing are phage, and most of them are uncharacterized. So there's a tremendous amount of viral diversity out there to capture using these new methods. And I didn't even touch on the RNA viruses today, so stay tuned for that. I'll wrap up just with a brief plug for our Microbiome and Public Health Center that I co-direct with Wendy Garrett at the Harvard School of Public Health. We do a variety of things to bring together the microbial community, research community at Harvard, in Boston, beyond. Some of what might be helpful for this group in particular is services that include stool and other microbial sample collection kits, including, apropos of the previous talk, culturable samples. So if you have a need to collect tens or hundreds or tens of thousands of stool samples, let us know. And we also offer a sort of bioinformatics consulting service that can help with standardized analyses of microbiome profiles. This is what we use to standardize our own microbiome epidemiology within the lab. So we have automated computational workflows that make all of this solid and reproducible. And then also of potential interest, keep an eye out for next May and June. We'll have our annual Center Symposium coming up, probably on May 18th. It's a little early, so we don't have a theme or a date yet, but mark your calendar. And then likewise, about a month later, we'll have our second annual week-long multiomics workshop hosted in Boston, doing a hands-on tutorial, several days worth of lessons around microbial community analysis generally. So I'll wrap up there because things are very short today. I didn't have time to call out the folks in the lab whose faces you saw who did all of this work. I tried to mention most of our collaborators as we went along, and I'm happy to use the last bit of time for any questions.